here,
Hello
not to attract
everybody,
your
I'm
attention.
Nikita
Okay.
Kohli. I am a
Oh,
student
it's a video.
graduated from the Master of Data Science program at Thompson Rivers University. Today I'm going to talk about leveraging lasso-based methodologies for enhanced SNP analysis in plant genomes. Today's agenda looks like this. Let's start with quickly introducing the important bioinformatics keywords and tools. Genome-wide association studies, abbreviated as GIWAS, aim to identify the relationship between genetic variations and physical traits. Single nucleotide polymorphisms or SNPs are the most abundant form of common simple DNA variants. There are various GIWAS software that are used for the analysis. Some popular ones include Blink, Tassel, Capit, etc. Blink is often viewed as a standard GIWAS method, and Tassel and Capit were developed for plant genomes. GIBOs have been criticized for missing important genetic markers, usually because the data may not fit well with the statistical models behind them. Additionally, the whole genome data typically have the large p small n problem, which makes detection of significant SNPs challenging. To solve this problem, we investigate how regression models can be used as an alternative to output significant genetic markers. Lasso and its variants have become popular methods for such tasks. Hence, the study evaluates two prominent lasso-based methods, namely Big Lasso and Iota Lasso. There are two main objectives of this research. First, assess the capability of Big Lasso and Iota Lasso to narrow down the feature set, providing a focused and relevant set of genetic markers. Second, contrast the outcomes of lasso-based methods with established reverse software such as Blink, Tassel, Capit, or FastLMM and see how much they agree with each other. In this study, the data from a model plant, Arabidopsis thaliana, is used. It is important to study plant genome due to the genetic differences between plants and humans. Plant genomes are more complex and larger in size or number and have a higher degree of homozygosity. This genome data has 1,307 samples with 2,14,051 SNPs or features. Two phenotypes from the phenotype data were selected, MCO5 the binary and FT10 the quantitative phenotype. Now let's understand the research methodology. The overall design of the study is to take in genotypic and phenotypic data and apply Big Lasso and Iota Lasso to identify a set of significant SNPs. The initial step is to test the two models on the datasets from their original papers. We have replicated the results and ensured that the programs can be running on our system. Then the two models were tested on Arabidopsis thaliana dataset. The output from Big Lasso and Iota Lasso are further tested and compared with GIVOS programs. Let's briefly discuss about the theory behind the methods used. Big Lasso employs advanced memory mapping techniques, leverages swift pathwise coordinate descent, warm start strategies, and support for parallel computing. This ensures efficient management of substantial datasets and boosts computational speeds, contributing to swift and optimized analysis. Iota Lasso integrates the alternating direction method of multipliers optimization algorithm to enhance Lasso's efficiency and accuracy in feature selection. It ensures automatic hyperparameter tuning specifically for adaptive lasso, contributing to improved model performance while maintaining computational efficiency. Output of Big Lasso and Iota Lasso are list of SNPs. The results of Big Lasso do not have rankings or significant values. The results of Iota Lasso come with coefficient values that can be used as the significance. GWAS typically provides SNPs with the associated p-values indicating the significance. To validate and test the usability of Big Lasso and IoT Lasso methods, we assess the performance of these methods on the 80 poly-DB dataset and compared them with Blink. For MCO5, the table showed the output SNPs from Big Lasso and their ranking in Blink. We can see that many are also top ranked in Blink, indicative of small p-values. This alignment emphasizes a high confidence level in associating these SNPs with a studied trait, strengthening the reliability of both models. IoT results did not yield any SNPs. Further investigation disclosed its effectiveness only with quantitative phenotypes, leading to the decision to exclusively employ IoT for quantitative phenotype analysis in our study. Big Lasso applied to FT10 phenotype involved data transformation to binary values due to its binary phenotype compatibility. An observed discrepancy between Big Lasso and Blink results hinted at the impact of the quantitative to binary conversion in the data preprocessing. For FT10 phenotype, we selected top 20 SNPs from IoT based on absolute coefficient values. Those were not top ranked SNPs for Blink, indicative large p-values. Further comparisons with Tassel, Capit, and FastLMM revealed similar results. Given the disparities between GWAS and lasso-based methods, an in-depth exploration was conducted to assess the linkage of SNPs to genes influencing the quantitative phenotype. Using TAIR website, 13 and 7 distinct genes related to growth and development were associated with SNPs reported by Big Lasso and IoT. This is evident from the tables in the slide. This supplementary analysis suggests that some SNPs potentially associated with the phenotype may be missed by the GWAS. Lasso-based approaches might complement traditional GWAS, revealing overlooked genomic regions and genes for further exploration. Now let's discuss the interpretation of the findings. Parameter settings may have influenced outcomes of no overlapping SNPs between Big Lasso and IoT. Good agreement between Big Lasso and Blink outputs in MCO5 highlights potential validation matrix, amplifies our confidence in the potential causal relationship with the trait, and opens the door for subsequent wet lab validation to verify their contribution to the trait. Both IoT and Big Lasso displayed limited overlap with conventional GWAS for FT10. Our validation of reported SNPs showed that some of the SNPs reported by Big Lasso and IoT pointed to the genes that are closely related to the phenotype. For Big Lasso, as we converted FT10, the quantitative phenotype, into binary values that may influence the results as well. Our study affirms Blink's robustness as a standard GWAS tool, providing a solid foundation for genetic analysis. Lasso-based methodologies emerge as compelling alternatives, particularly in scenarios with large p small n datasets. Their efficacy in identifying significant SNPs contributes to streamlined and insightful genetic research. A few potential future directions are to investigate other lasso-derived methodologies such as group lasso and ridge regression to expand the range of analyzed techniques. Conduct a detailed study on the impact of parameters in lasso-based methods to enhance the understanding and optimize outcomes. Extend the study beyond Arabidopsis thaliana to encompass various plant genomes, enhancing the universality and applicability of the findings. Investigate methods for combining and ranking SNPs outputs from various techniques, aiming to enhance accuracy and confidence in reported SNPs. Finally, develop a pipeline that synthesizes and reports significant SNP sets in a customized and reliable manner, providing researchers with high confidence in the association and predictive potential for phenotypes. Now, I would like to thank the authors Dr. Nisha, Dr. Yue, Dr. Randy, and Dr. Yan for their work and constant support and great guidance. Thank you for listening to my presentation. If you have any questions, please contact Dr. Yan Yan at the email address on this slide.